CAR T cell therapy assignment. It's a drawing assignment. There are some drawings in here that I describe as we hit them. And so it's a type of cancer therapy we'll get into. It's used to treat blood cancers, so lymphomas, leukemias, melanomas, and it's being very successful. And we'll see that it has these parts. Before we get into the parts of it, this is not a course in immunology, so we want to go very gentle on this, but it's important to introduce as the example what normally happens. And so a lot of times, we talked about in our course, cancer cells should start growing out of control. Cancer cells should go through apoptosis, and they do. But there's another level of natural healthy surveillance that occurs, and it is that our immune system can recognize cells that are out of control, and it can kill those cells, and it oftentimes does that, or I will say typically does that, by causing that target cell to go through apoptosis. So let's take a look at that just to set the stage for that and the cells that are involved, okay? So here is a situation, and we'll just talk through it. And so we've got just the storyline down here. There we go. Just a storyline down here. So first, this is the T cell that we're going to be talking about. And it binds to this cancer cell. So it's recognizing a normal, healthy cell, and but the cell is growing out of control, right? And so it's going to bind to that and get activated. And it gets activated by requiring multiple receptors to get activated on the target cell and also from other sources. And so this is a helper cell that's going to recognize that problem cell and say, yeah, get going on that. So releasing signaling molecules that bind to this. And these get integrated, and that's what we're going to be talking about. The whole story is focused on this cell and how this cell has checks and balances, because this is a dangerous game to play, because we don't want an autoimmune response. We don't want the immune system attacking our normal cells, right? That's what happens in autoimmunity. So we're saying, be careful. Watch for your own body making a dangerous cell and be ready to encourage it and drive it toward cell death. But be careful. Require multiple signaling, so that would be this integration and crosstalk, right? That's what we're talking about. So it's a fantastic example of the need for that integration, crosstalk, multiple pathways. And then that's going to respond, it's going to kill this cell, and importantly, it's also going to be stimulated to proliferate and not die. So remember that we learned that these immune system cells are constantly made by our bone marrow because they only live a few months, and if they're not activated, they die. And that's part of our immune system's ongoing sort of immune surveillance strategy, is keep things fresh and new, keep looking for trouble. And if you find trouble, have those cells proliferate, so cell cycle response. So this cell is going to make a cell cycle response as well as trying to kill that cell, or encourage that cell to die, rather. It's going to make a cell cycle response, and it's not going to die. So, really important stuff happening here from our course, okay? And so here's the thing. Scientists have found target molecules on cancer genes that some cancer patients, simply aren't recognizing and killing. So the plan in CAR T cell therapy is, let's take these cells out of the body, blood, removing blood cells like that, like donating blood, getting these cells, and making a super receptor, having a super receptor gene we can put in that'll make a super receptor that combines all these into one. So instead of requiring that integration and crosstalk and care, we just make that thing go after it with a great and easy activation. The anxiety is we might get it, to kill some of our off-target cells. That's an autoimmune response, right? Not hitting the right target like that. That's a big anxiety. But the idea is, let's just crank it up and fight this. And, it's kind of a last chance fight against this mean cancer like that, right? So that's described there. That's the normal therapy, and that's the strategy. It's the whole strategy. Okay, how does it get done, all right? So the focus is on that cell and those receptors. That's it, okay? So just to show you the treatment plan, we're not dwelling on this, but what does it look like when it's cancer treatment? And so here's a simple view of it. The patient has blood cells withdrawn. These T cells are identified. A transgene is inserted to them that's going to code for this super receptor, and they're put back into the patient's blood. Oh, they're expanded. More proliferation occurs in the patient, not because of activation, like I just described, but just because let's get a bunch of these cells back into the patient, and then they just go back in the patient, through an IV drip like that. Okay, and I should also mention that this is quite successful. These patients are. This is a last resort strategy. So they've had cancer, and it's a tough cancer, and it's a blood cancer, and lots of these patients get better in 2 weeks, and lots of them stay better for years. So, it, of course, doesn't work every time, every place. It's also very expensive, so it's a last resort strategy. But at the end, we'll talk about how this is a new wave of cancer treatment style, and it'll simply be used for other types of diseases and health issues in humans. So here's just a longer description, longer image of what happened. Same image as the previous one. It's called an autologous transplant. A heterologous transplant is when you get a kidney transplant from another person. So this is not a heterologous transplant. These are your cells, the patient's cells. They have been made into trans cells. This is a trans cell that's going back into the organ because that's going to be a trans gene that was built in the lab. And that's just available to the doctors, right? A company built those transgenes that sells them to the doctors and scientists like that, right? So that's a treatment plan. Great. You're not going to be tested on that. This is what we're interested in learning, right? So we set the stage for this example. We want to be able to draw that cell and show how it's changing in response to this signal, okay? So the first one is building the transgene and expressing it, okay? So there's the idea. We need to make 2 proteins out of 1. How are we going to do that? And a key concept is that here is the way to create new proteins is not by purifying the proteins. We don't do that. It was never done. Instead, the genes are isolated and manipulated. And we know how to do that. And we're just going to start drawing it right here, okay? So we're going to have, and I'll just draw them out, right? There's going to be 4 genes that come together. Whoa! 4 genes come together. 4 different genes, and again, you need to label all the parts of these, so show that you understand the parts we want to get together. And there you go, 4 genes. And what are they going to do? Well, they're all normally expressed in T cells for the cis regulatory region, right? That's not what we're changing here. We want to use any one of these genes for the cis regulatory region because it's going to have a transcription factor that's going to activate it in those T cells. We're going to put this transgene into T cells, and we want it to be expressed in T cells. These are expressed in T cells. The 4th one is not. That's okay. Don't worry about that. The 4th one is not. It's an antibody gene. And it's not expressed in T cells, sorry. But these 3 normally are, so any of these 3, and just pick one of those. So this is not a story about the complexity of the cis regulatory region. We just want to get it expressed all the time, and that's what these receptors are in the T cell. Then the next thing is, what are we going to change in these genes? And the answer is the coding region. And we're just picking this one, and so there's its coding region. And what we're going to do is we're just going to start replacing parts of its coding region with parts of these other proteins. So we want to use this as the activation region inside the cell, we'll say. So we just add some of the blue one. So it's got that activation region. So it's going to activate that pathway of signal transduction. And then we're just going to add this part of it too, like that. And then this antibody, that's the extracellular part. That's the part that's going to bind to the cancer cell. So we've got this, and it's going to make a protein, and we could draw the protein, and the protein looks like this. It's going to have this part outside that's going to bind to the cancer cell, and then it's going to activate this. There's a part that's just called the hinge that comes from one of these, and just make it different, but it comes from one of those. And then inside, we have these 3 parts that are all part of the same protein now, and each one of them is going to activate one of those important signal transduction pathways. One, like that. So this one is going to activate the pathways that are normally activated by this one. Maybe there's more than one pathway, right? This one's going to activate those, and they're all integrated. They're all put into one super receptor, and it's called a CAR receptor, and that stands for chimeric. And I think we've used that term before. Chimeric means the kind of a hybrid kind of monster. Like in Greek mythology, there would be like a horse with like a lion's head and bird's wings, and that's a chimera. And these are chimera, and then chimeric antigen, and an antigen is what's being recognized on the cancer cell. And the antibody's doing that. So those are terms from immunology. Great, fine. And then it's a receptor. So CAR, chimeric antigen receptor, and we made it. It's a supercharged receptor protein. And you can see what we've done there. We don't need the three different receptors going. Just activate that one and get going. Like that. Make a big cell response. So you should be able to draw that, right? Take time to draw that. That's a first drawing assignment, and include the labels: cis regulatory region, transcription unit coding region, and show how you take in your drawing. Show how you're taking parts of the coding regions of these different genes and putting them all into one, and that you're using that original cis regulatory region because that's going to be expressed like that. So it's a drawing, and it's just very satisfying. We've done these kind of drawings before. It's just dealing with the coding regions because we don't want to change the cis regulatory region. We just want to change the product that's going to get made. So draw and label the CAR gene and the CAR protein product like I just did. Make it a little bit better drawing, right? I'm making dry drawings, and I'll continue to make the drawings in this pretty quickly, but fill them in. They don't have to be elegant. You don't have to be an artist, but make it a good quality learning experience to learn this material, and then also to see it in your mind. So draw it big, label it big enough so you can see it in your mind when you recall it. You're like, oh, I can read the drawing. I see it like that, right? And you might learn differently, but try this or try something similar to this, okay? So that's the first thing, is just what did they make in a lab to put into the cells? And then what does it mean when we integrate? What are the signaling pathways that those three receptors normally use that get all put together? Okay, so the receptors are not the kind of receptors that we've learned the most about in our course. The three receptors are not receptor tyrosine kinases or G protein coupled receptors. They're in that last group of receptors where there are different sets. So, for example, one is a non-receptor tyrosine kinase like JAK-STAT, but it's not JAK-STAT. So we're not going to learn the names of these receptors because they're in that unique category. But what we're going to do is we're going to see the signal transduction pathways that they activate. So here's a pretty first simplified example, and we're talking about these three receptors, which again are not receptor tyrosine kinases or G protein coupled receptors. That's fine. And they're going to activate these signal transduction pathways, and some of them are going to activate transcription factors and cause genomic responses. And those include things like this cell should continue to live, this cell should proliferate, this cell should make other secreted factors that we'll talk about that will help activate the immune system. Okay, so let's just look in our learning about what these receptors activate as signal transduction pathways. So there it's activating RAS and MEK-ERK-ELK, RAS, RAF, MEK-ERK-ELK, and so this is getting transcribed. It's activating c-Jun. It's doing that by activating the expression of Jun. This is Jun-FOS. Learn a little bit about that. That gets activated by RAS and that RAS pathway. These are other pathways we didn't learn about, but that's this receptor. And we'd kind of predict that's probably going to be involved in like cell proliferation. Yeah, maybe cell survival, like that, right? Could be other stuff too, because lots of target genes. But we know that receptor. We don't know that receptor, but we know that signal transduction pathway. And that's pretty common. There are lots of receptors, lots of pathways, and we've learned a lot about both. So in a given example, we'll probably know a fair amount. Then here's this pathway, right? This is that PI3 kinase is going to activate a pathway so there's PI3 kinase right there, right? That's involved, even though we learned it's often involved in receptor tyrosine kinases pathway. It's a different receptor using PI3 kinase, getting that PIP3 to activate AKT, and AKT can help activate changes in gene expression, okay? And then here's phospholipase C, also being activated like that. And so familiar pathways into the cell to activate cellular responses, including genomic and non-genomic responses. The genomic responses are, kind of especially emphasized here. What's going on here? Well, this receptor especially activates a pathway we didn't learn anything about. That's fine. There are more of those, but we understand what's going on, right? Binding to, extracellular molecule that activates the receptor and it gets communicated into the cell through a signal transduction pathway. Here's a more complicated one showing those three receptors now bundled into the one transgene CAR gene and CAR protein. So we have that extracellular binding domain made up of the antibody, then that transmembrane hinge or linker region like that, and then the three domains of these three receptor proteins all put together in one, like that. And so just to show you again, here's that PI3 kinase pathway to activate AKT. Here's this is actually including a different kind of MAP kinases. So remember we learned that MAC, RAF, MEC, ERK are all these MAP kinases. So here's a version of that MEC going down a different pathway to activate changes. And they'll probably miss some. Here is the phospholipase C action going on. Here is the classic, it's not RAS, it doesn't show RAS on there. Oh, there's RAS, RAF, MERK, ELK, like that, right? So familiar pathways, right, that are activated now all together. One receptor, no longer requiring those checks and balances because it does it all, like that. And so, just doing the kind of stuff that we're familiar with into the cell, right? Because it's a lot of signaling pathways all bundled together, right? So draw and label a very simple drawing of the CAR protein activating multiple signal transduction pathways. So you don't need to draw out the details, but I would say just based on your memory from the first exam, draw out your memory of the pathway. It was on the exam, to activate AKT, and then this phospholipase C pathway, and the RAS, like that. Draw that out. Draw a picture like this with the CAR protein now, right? Just to show that, to get that feeling of, oh, wow, I see what the scientists did to help the doctors. They made a transgene that makes this super receptor CAR protein. They modified the gene. They didn't mess around with the proteins. They modified the genes, put them all together, and they put them together, of course, in this specific way by altering their coding regions, like that, taking portions of the activation part of their coding regions to make those parts of those proteins. Really just amazing, from what we learned in our course. It's just amazing strategy, right? Like that, okay? So draw that out. Again, keep it simple, but, show that you understand in a drawing and show yourself something to remember on our exam. Because I could say, give examples of signal transduction pathways that get activated in a CAR T cell. So here's the bottom half of that, just to show you that lots of these signals are going into the nucleus to alter gene expression. This is going to be an immediate response, right? The cell's going to be activated. We're going to see there's some non-genomic responses very fast, and then some longer-term responses, changing gene expression. With each change of the cell gets activated, so it's going to change, and it's going to increase its metabolism. It's going to change and increase the types of genes it's producing. It's now got a job to do. And then it's also going to proliferate and it's going to survive. So those are going to be some long-term changes. It's going to stick around for years like that, right? So long-term changes, including changes in transcription factors like that, okay? All right, so that's the second one. The first one, is what's going on, and, we got that, right? And then how to build these. Then what does that mean? What does it look like? And then here's the third one that is important for us. What are the responses? What type of cellular responses does that CAR T cell make? And the answer is it makes the same responses as it would in the normal cell, right? It's going to do the same things as it would have normally done in the patient. We're just putting them all together to get it supercharged, to get more poised to do it in response to finding that cancer cell. So, but it's going to make these same responses, and that's this line drawing this way, right? It's going to make these same responses, so it's going to make a different kind of cell and more of them, and it won't die, and it will be more poised to just kill, and that's more cancer cells in the future, right? Like that. And it might be a few days or a few years like that. Really, really amazing stuff going on, right? All right, so cool. So what are the types of responses that the normal cell does and that this CAR T cell does with enthusiasm, right? Like that, okay? So again, I guess I got to go forward a little bit further. Sorry, the genomic and non-genomic cellular responses. Okay, so we're going to learn these, and I didn't have another one, but there are like that. But these are really good ones. So non-genomic responses means we don't even need to make new genes expression. We don't have to activate genes, and so these are some things that are happening right away. And so there are going to be changes in the cytoskeleton. And I've got an image of what that looks like. Okay, so we'll talk through these. Okay, and then there's going to be changes in the types of proteins that are getting released from the cell. Some of them are going to be proteins that are going to encourage that target cell, that cancer cell, to go through apoptosis. Others are going to be released and bind to other T cells, different kinds of T cells, to have them help with this job and help kill that target cell. Okay, so the first group, okay, the changes in the cytoskeleton, and so they undergo rapid cytoskeleton remodeling. I'll just show you what it looks like here. So here it is getting activated, and what I say is the cell changes its shape to hug this. This is the cancer cell, and it's going to hug it. And what is that going to mean? It's not shown here, but when this starts wrapping around the cancer cell, it's going to activate more of those receptors, right? Because they're going to make more contact like that. Then it's also making this space, a special space right here. These cells are very close to each other, and you can see it's going to start releasing factors. And you're going to release them right here. Not going to release them over here. And so what's going to happen is we have a change in cell shape, and then a change of what's happening inside the cell. Like that, okay? So the changes in cell shape are depicted here by these red, and these are actin microfilaments. And when we think of microfilaments in the cytoskeleton, we think about the cell shape changing, and the cell shape change here is, and it's not an official term, what we say, it's what makes the this CAR T cell hug the cancer. And it's just going to it's going to increase the activation response. It's like, yes, 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 right? More receptors getting activated, more cellular responses. So it's like a positive feedback loop for the T cell. Yes, I'm doing the right thing. I'm getting further activated. And then it makes this close contact where it can release these factors and encourage that cell to go through programmed cell death. So that's the actin microfilaments. And then these are, and that's the red. So the active microfilaments are the red molecules right there, and they're changing the shape, which is what the microfilaments do for the cytoskeleton. primarily. And then here are the microtubules. And the microtubules are like train tracks, and they're involved in moving molecules around. And we get a reorganization of the microtubules to put them in position here, where we're going to be making railroad tracks to deliver new material up here. And let's get this job done. So really a beautiful cell biology image like that, okay? So the CAR T cells undergo rapid cytoskeletal remodeling upon binding to the target cell. And actin polymerization, this reorganization is critical for establishing the immune synapse. That's that gap, and we don't need to know that term, allowing the cell to spread over the target and form a stable contact. Then within a minute or two of contact, the microtubules reorganize, shifting everything towards secreting molecules, bringing molecules to that area. That's that connection area. So there's some terms here like that immunological synapse. That's obviously not a term we need to dive into for our course. It's not a course in immunology. It's really that visual. That's just a beautiful thing. And it's a great example of a non-genomic response. We don't need to wait around for new gene expression for this, right? And so it's just for our whole course, it's a really great example of a cell response. This is a cell response responding because receptors have been activated, signal transduction pathways are occurring, and it's going to respond. And the response is going to be, I'm going to wrap myself around you and move stuff toward you, like that. Here's just a microscopic image of what that looks like. The green are the active microfilaments, and you can see this one has made the best contact already. The rest are still rounded up, but as they flatten out, they're making better contact, right? And that's the first set, the changes in the cytoskeleton. Just a beautiful, beautiful image for you to remember for the rest of your life. Just fantastic. And that's what the T cells do in our normal body. This is a normal cellular response. We've just increased it. Increase the enthusiasm to make that response by putting all those receptors together. But this is what this could be a normal T cell responding to a troubled cell in your body, like that. So then, the T cell has already got some proteins, and I don't really care if you learn these proteins, but we should be able to include them in our drawing. And so this is what they do. Okay, so just put them in this drawing. And I'll erase this to draw this. So in this gap, we're seeing these released. And that's what these molecules do. And some of them make channels in that healthy living cell. And others of them go in through those holes. And these activate caspases. Right? This cell is still going to kill itself. And this cell is the T cell is saying, yeah, you are, right? I'm just going to skip over all the regulation, the BCL2. I don't care about that, right? I don't care if you have a lot of BCL2, right? You might have had a translocation and you have a lot of BCL2 in you, and you're truly trying to repress that, right? Trying to keep those off, and I'm just going to activate them. I'm just going to bypass all the regulation. I'm going to activate them. So it's just a brutal, aggressive move. This is not killing the cell directly. It's just going to activate apoptosis, because remember, the caspases are those action proteins, and this thing is going to start flipping those phosphatidylserines out. It's going to start destroying its nucleus. It's going to start making its mitochondria leaky. It's going to degrade its DNA. It's going to attract through those phosphatidylserines another cell to come up here and just phagocytize it and wipe it out. So we don't need to remember the name porphyrin or granzymes. Fine, those are the molecules that make the hold and go in and activate the kinases. You can learn them if you want, but I'm not going to specifically ask you for the names. This is the one that makes holes. This is the one that goes in. Just fantastic, and they're ready to go, right? They're just ready to go, right? So draw and label that. Draw and label what I just skipped out here, and just make it bigger and better by leaving out the stuff that you don't want to learn about, and putting in these that you do want to learn about. And you can activate it. You can say right in there in your drawing, activate caspases, even if you don't learn the word granzyme, and you could put holes, like that. If you don't want to learn perform, that's great. It's good to learn microfilaments and microtubules. Those are really similar names, but they do very different jobs. Microfilaments change the shape of the cell. Microtubules move stuff around in the cell. So whenever I draw microtubules, I just like to make them look like railroad tracks. That's how I draw railroad tracks really quickly. That just remind myself that's going to be moving stuff around primarily like that. Okay? So make a drawing of that. Oh, man, it's just some fantastic thing, right? And then the last non-genomic one we want to talk about is, well, let's just go through this a little bit. So the fifth one is that the CAR T cells are also ready to release signaling molecules. And these are molecules that activate other T cells. So activate other T cells, not more of the CAR T cells, but other T cells that do other jobs. For example, phagocytosis, or helping to stimulate other cells in the immune system to help participate in this process, like that, yeah? And these secretory factors in immunology are called cytokines and chemokines. Those are just a general name for secreted signaling factors that activate and regulate the immune system. We don't need to learn that term, but we want to learn the term that this T cell or this CAR T cell is ready right now to release molecules to help, other cells, other immune system cells, T cells, get involved in this fight. Yeah. So then genomic responses, right? So those are really beautiful non-genomic responses because they make sense. Let's wrap around this cell. Let's release these factors. Let's release other factors to help other cells get involved, right? It's kind of at the front end. Let's go. It happens fast, and that's a good example for non-genomic responses. So then genomic responses, that means we got to activate new gene expression. We got to activate transcription factors. And what we're going to do, these are good answers for the cell. When a CAR T cell gets activated, the transcription factors activate expression of genes that do what? Stimulate the cell cycle. So what does that mean? Go through start. Just go through start. Our very familiar thing. The T cell is floating around in the blood. It's not activated. It's going to hang out for a few months and die. It's not going to be dividing. It's just going to hang out. But when it gets activated, it goes and gets a big green light to make more of itself. Then it's going to die if it's not activated. When it gets activated, don't die. So it really flips the plan to decrease apoptosis. That's going to be new gene expression, changing the plan, like that. Then it's going to have to do that by increasing the metabolism. We got to get this thing going. It was inactive. It's waiting. It's floating. It's not using up a lot of body resources because it hasn't been, activated. But as soon as it gets activated, the body's like, let's use this cell. It's activated. Let's keep it. Let's make more of it. Let's give it a lot of energy like that, and then that initial non-genomic response of releasing things and doing things. Well, it's got to back that up with continue. Let's make sure we activate those genes they're already on, but let's crank them up like that, right? Okay, so great, and this is just listing pathways to transcription factor activation. And these pathways induce transcription of essential genes, leading to cytokine production, rapid cell cycle production, cell cycle expansion and progress, and release of cytotoxic molecules like that. So let's just do more of that and get ready. And that's that. And I'm not asking you to draw a picture of it, but it'd be really good to learn that list. And if you wanted to incorporate it into a picture, great. Okay? Then our last thing is, this is a different kind of cancer therapy, right? So there's surgery, there's radiation therapy. Let's try to kill that tumor by exposing it to, mutation-inducing X-rays, like that. Or there's chemotherapy. Let's give this patient a drug to fight that cancer, like that. So these types of treatments are, let's use immunotherapy. And immunotherapy means let's get that immune system going. And there's a variety of ways people are having success doing that to fight cancer. And then it's also this example of cellular therapy. Let's get the cells in the body to do this job. like that. That's a new strategy. So we want to focus on that. So there again is that original, well, this is the normal cell. And we've talked about this is just going to do its normal cell thing. It's going to get activated here. So it's going to get activated. And we've just made the activation simpler. Put all these together in one transgene, the CAR gene that makes the CAR protein, to make it a super receptor that allows it to get activated right away. And it's just going to then do all these responses are totally normal. And that's a key concept. We're making a supercell, but only in its detection of the cancer cell. The other things, all the hugging and all the proliferation, those are totally normal. So it's immunotherapy. Let's get this patient to get going. Let's get their own immune systems. Get them going. Get them more activated. Here's just a simple drawing, just to show you again. That's the setup, right? Really, the bottom line is we've got this fancy receptor, this super receptor, and it's going to bind to targets on those cancer cells and kill them. And it's going to kill them by talking the cancer cells into killing themselves. Yeah, like that. And remember, cancer cells are they're not doing that, right? They've decreased their willingness to go through programmed cell death. And you just seen how it just, so aggressive, and so effective and so nice in matching up with the learning we're doing in our course. You should just be nodding your head and being like, yeah, I can draw that stuff out. So it's a type of immunotherapy, and it's especially used in blood cancers so far. And it just has great success rates, given that these patients, this is their last hope for treatment. And in lots and lots of folks, it's very effective, very quickly effective, and long-term effectiveness. Okay, so that's immunotherapy. And I wanted to show you this because there's a variety of immunotherapies, and we're just going to talk about 4. So these are all about, let's get that immune system going to fight cancer. And immunotherapy could be used in other diseases as well. Let's see if we can use the immune system to help fight that disease. So let's just talk through 4 of these. The first one, we already learned about this. Our example is from the first exam, cetuximab. And that binds that ERB-B oncogene, and we should be ready to describe that on this exam. So that's the example where you better go back and find your knowledge about cetuximab and be ready, because I could say, give me, 3 examples of immunotherapy, and you'd be like, wow, I could use the cetuximab. And you don't have to learn the word monoclonal antibodies, because that's from immunology, but it's a treatment that uses those. So cetuximab is used to treat patients that have that ERB-B oncogene. Then the second one is we're just going to look at news headlines. Just the idea that there are these vaccines. And the vaccines are these modern style mRNA vaccines. And I'll just mention this once. The federal government gave 2 biotech companies billions of dollars to make mRNA vaccines against COVID. When the government gave them that money, even though it was early in the pandemic, those companies had already started testing these vaccines, because there is a very long history of companies and scientists working on developing the idea of an mRNA vaccine. And now, if you look at the companies of those, at the website of those companies, they've used, the resources. I don't want to say they use the government money for COVID. They use the resources and, investments to just start making many, many other mRNA vaccines. And so these are mRNA vaccines that are vaccines against specific cancers. And they're being tested. And right now they're being tested especially and this is just news, they're being tested especially for trying to help from somebody who is going to have a relapse. So they might not be the first sign of, fighting. You'll never get cancer, you got a vaccine. But rather, you've had cancer, and we want to try to help you from having that one come back. Okay? So that's all we're going to say about that. Just vaccines, but include that because it's a kind of immunotherapy, because we're using the patient's immune system, right? Like that, okay? And of course, there are other examples, but that's it. Then there's CAR. We just talked about CAR, our third one, and we just want to talk a little bit about a fourth one, and you'll see why. And the fourth one is this one that's called adoptive immunotherapy. And this is a very simple story of it. There is a patient who has a tumor, and the tumor is removed by surgery, and now scientists and doctors will find the T cells that are in that tumor. So these are the patient's own T cells. They haven't changed at all. But if these T cells are in there, they're probably trying to fight that cancer. So after surgery, let's take that tumor and not put it in the trash can. Let's get the immune system T cells out of it. Let's increase their numbers in the dish and put them back in the patient. Get those fighters back in there. So it's a type of adoptive immunotherapy. It's the patient's own cells. It's just like you should have more of your cells should be fighting this cancer now and in the future. So we're just going to take the tumor, find those immune system cells, give you more of them, and give you those same cells back because they're your good cells. And maybe you have a weak immune system, whatever, but you're trying, and we're going to try to help. So that's this adoptive immunotherapy, and it's a cancer treatment that removes the patient's own immune cells. And it removes them as part of that surgery. They're in the tumor, like that. So really satisfying story, like that for us. Very simple. We don't have to draw it. And then here I'm just showing off on the side here. The bigger figure shows that they've taken that basic strategy and they're doing this. Wow, now we're going to start manipulating things. Now that we have it out of the body, we can manipulate them. So do more to help get these guys activated, right? So CAR T-cell therapy and adoptive immunotherapy are two types of examples of immunotherapy, right, that are also called cellular therapy. We're going to take the cells, and that makes them different than surgery, radiation therapy, and especially chemotherapy. We're not going to give the patient a drug. We're going to give them a cell. And now that they got going on this, they just keep going. They just improve that. They've gone through several generations of these CAR genes, making them better every time. Better switching, better at targeting the cancer and avoiding the healthy cells throughout the body, right? No autoimmune diseases like that. Yeah, just better like that. And it's a big new field, and we want to learn about that. And so, for our test, we could say, give me 3 examples of immunotherapy. And if I were you, I'd pick cetuximab, CAR, and vaccines. This is some new learning. You can pick that one too, adoptive immunotherapy. But, right, we just learned about CARs, about them, about cetuximab, and the vaccines is just to describe. You can make a vaccine to fight certain cancers. Yeah, like that. Then I also want to show you that of these examples, some of them are cellular therapy or cell therapy. So I could say, tell me about the 2, right? 2-point question. What are the 2 types of cell therapy we've learned? CAR T cells, CAR T-cell therapy, and adoptive immunotherapy. Great, okay? So then just go back, right? We had to start out looking at the normal story in CAR T-cells, and then we really want to focus on this. I guess the numbering is a little messed up here, but, draw a picture of that, draw a picture of that, draw a picture of that, to memorize it, learn it by not just looking at a list, right? But actually looking at an image. And it's an image you've drawn to make it most efficient. It doesn't have other labels in it like my figures always do, right? They got extra labels in them like that from the book or from the website where I found them. Make one for you, the learning we did, right? And then, no drawings about this, but some learning about that as well, right? Okay? And so we're good. All right, thank you. And that's due, coming up as already on our announcements. All right. So thanks a lot.